Greetings and welcome to the NanoViricides Webcast. It is now my pleasure to introduce your host, Anil Dewan, President and Chairman of the Board. Please go ahead, sir.
Good morning, everybody. So I am the President and Chairman of NanoViricides. It's a public company. It was formed as a public company in 2005. The symbol is NNVC, Nano Victor Charlie, and we are trading on NYSE American. We have been developing drugs with using a completely novel technology that I will touch on a little bit, and which is basically like a Venus fly trap designed for the virus to attract the virus, enclose it, and destroy it. And it can do it inside the body. Every time I talk to scientists, they say that cannot be done, that it is just science fiction. But we have actually done it. We have also shown in electron micrographs that it does work the way we are saying it does work. And now we have started progressing towards our first IND application for the treatment of shingles rash using a skin cream based on our technology to control the virus that causes shingles rash and chickenpox, VZV, varicella zoster virus. So I'll give you an update on that. Along the way, as we were preparing the IND, came this startling news out of China in December, towards the end of December, that something was happening and people were dying. And we looked at it. We know, of course, about the SARS-CoV that had happened in 2002 and MERS coronavirus that had happened in 2014. And we had actually worked on those in terms of developing some possible ligands for those at those times. In 2002, of course, the company was not NanoViricides. And we had some structures because of that in our library. So we decided to take a quick look at that. And we found that we have something that can make a difference on the COVID-19, which has become a pandemic now. At the time we started working on it, it was simply, we don't really know where it is going to take us, was the status at that time. And we are very happy that we have engaged into this war against the virus. And I believe I cannot show you any data yet because it's still under curation in our R&D team. But I'm very happy to say that we do have possible drug candidates that can make a difference. So with that, as an introduction, I will start going through the slideshow. Next slide is the disclosure statement, and it applies to everything I said. And when I open my talk in front of live audiences, I usually say that what this statement from SEC says, that basically I am allowed to lie and you have to decide which one is not lie. I'm a scientist. I don't like to lie, obviously, and I like to base things on data. At the same time, scientists are dreamers, so they extend data and to try to see what possibilities it might bring, and that I do too. And that's why a lot of times what I describe is considered science fiction, even by really well-known scientists. Next slide, please. Okay. So in terms of SARS coronavirus drug development, we were very lucky. What happened is that very quickly the Chinese scientists discovered and published that the new coronavirus was also binding to the same cellular receptor ACE2, ACE2, angiotensin converting enzyme 2, which is actually involved in blood pressure maintenance. That receptor, same as what the original SARS coronavirus was binding to. And that gave us, like I said, we had worked on these things before, so that gave us a leg up. We looked at some of the structures that we had. We did some molecular modeling studies, and we found that we have some ligands that can bind to this virus. And once we have ligands that can bind to the virus at the site that we desire, which is the site by which the virus actually binds to the receptor, because that gives you an ability that the virus will not escape the drug. And the reason why we say that is that no matter how much a virus changes genomically, it ends up binding to the same receptor with the same footprint. And so if you are using that footprint and mimicking that receptor's footprint where the virus is binding, then you are pretty certain that no matter how much the virus mutates, it's still going to continue to bind to your drug. And if your drug is successful beyond that in encapsulating the virus, it is going to continue to do that. So the escape of viruses by mutations, which is the dreaded thing for all the approaches that are currently being talked about, the antibody approach, is very sensitive to escape viruses because antibodies are extremely specific. The vaccine approach is very sensitive. Vaccines are, of course, prophylactic and won't be there in time anyways, but it is still, as you know, every year new influenza viruses vaccines have to be combined and manufactured, and that's because the viruses change over time. And because of that, it's very, very important to develop something where the viruses will not flip away from. And that's what we believe we have done. One of the reasons that we can say that we have achieved that goal is by trying this same drug that we designed against one virus in that class against different viruses in the same class. If it binds to those as well and is effective against those as well, then we think that we are very close to the goal. And that we have demonstrated already in the case of the herpes virus family. The shingles virus drug that we are now taking into clinical trials, IND is being prepared and is going through consultation with consultants. That was actually designed to work against herpes simplex virus 1, the virus that causes cold sores, and it was also shown in our lab to be effective against herpes simplex virus 2, the virus that causes genital ulcers. So we expanded the pipeline without doing any new work, and we are adding more and more indications because of that. It is a broad spectrum drug that is attacking at least these 3 different herpes viruses. There are more herpes viruses in the family that we have not tested against. So the same kind of thing we are doing against the SARS coronavirus as well. The success of the herpesicide program is because we designed it against the known herpes virus entry mediator receptor of HSV, herpes simplex 1 and herpes simplex 2. VZV was not even known whether it uses that entry mediator or not as a receptor. So we were surprised actually when we found in our studies in the lab that it worked against VZV as well, and that gave more credence to the hypothesis that we were actually developing relatively broad spectrum drugs by the way that we design ligands and by the way the polymer is made and designed to attack the virus. So that's where we are at, and we believe that we have done the same kind of a thing with the SARS coronavirus drug program that we have right now. This SARS coronavirus, as you know, has become an economic nightmare for the entire world. In the US alone, the government has already put in more than $2.5 trillion in supporting the economy and trying to develop approaches against the virus and supporting healthcare, and that's not the end of it. We are going to see economic slowdown. That is going to be lasting for a while. We are going to see healthcare issues that will continue because a lot of elective issues and surgeries and things like that, a lot of normal procedures that we do for health maintenance are being held back. And once hopefully that crisis is over, all those are going to overflow into the healthcare system. So all over the place, we have a tremendous amount of impact by this one little virus, and we believe that we have the only possible solution, that is a drug. When people get sick, you need to use a drug. For preventing people from getting sick, you can do social distancing, you can do vaccines if they become available in time, which in this case they are not and will not be, no matter how much people say that they will be ready, etc., etc. Because developing a vaccine to be safe in 1 billion people is a very different ball game from developing a drug that needs to be safe in only the 10,000 patients, 100,000 patients. So those are very, very different statistics. And you have to have a vaccine that is very, very safe, and that requires a lot of work. So going back to where I was, that again, vaccines and antibodies, they are very sensitive to viral mutations, and viruses escape them readily. In this case, SARS-CoV-2 also, a lot of virus mutations have happened. There are many different modalities that we are discovering that this virus is causing in different patients, different way that it is showing up. Some patients are dying, similar people are not dying, and all that kind of thing. So there may be differences between the circulating viruses as well, and that can cause differences to drugs. So there is an important thing that needs to be done is to develop a broad spectrum coronavirus drug. And that's what we believe we are doing by mimicking the ACE2 receptor to begin with. And we are also testing that drug against other viruses that do not use ACE2 in the SARS coronavirus family, and we have found success in that as well for a different reason, because those receptors belong to the superfamily that ACE2 belongs to. So next slide. To go to the next slide, before that, I want to also tell you that we are already ahead in terms of cGMP manufacturing facility. Nanomedicines are extremely difficult to manufacture. It's a known risk. And so we went ahead and built our own manufacturing facility as well as analytical capabilities by nanomedicine characterization facility way back in 2015. That is fully operational now. We have already made cGMP quality materials for the clinical studies that we are going to be for shingles, for the preclinical program going into the IND filing, the tox package studies. Those were cGMP compliant materials that we had made in kilogram quantities. Now, a kilogram, if a drug is highly active, can treat 1,000 patients. If you have to give it daily for 5 days, you are going to treat 200 patients. That's one batch. And we can make one batch per 3 weeks or 4 weeks. So we can definitely supply the material that will be needed for treating the hospitalized patients from the SARS coronavirus epidemic from our own facility. There is not going to be a need for a new manufacturing facility. Of course, we will have to work all around the clock, and we are already working around the clock. That is known in this business. Anybody who is working on the coronavirus currently is working very, very hard. And we appreciate that. We are not the only ones. So going to this slide, we are doing a biomimetic technology, and because of that, we have already developed drug candidates. That's a major milestone. If you look at antibodies, they are still discovering antibodies. They are going to put them into trials. If you look at vaccines, mRNA vaccines, DNA vaccines, they have candidate vaccines. They're putting into next step, and they're going to go further with that, and so on and so forth. We already have a drug candidate that works in cell cultures. We are going to go into animal studies. We are going to, side by side, we'll do the safety toxicology. We will do an accelerated safety toxicology program for this, and that will take us to the ability of going into first-in-human studies. Now, these are not going to be standard randomized clinical trials, beginning because of the pandemic nature and because of where the epidemic stands right now. We'll be beginning with severe or moderately severe disease patients and just a few of them to see how it works. It's sort of like what happens in cancer drug development and the early days of cancer drug development, when even a drug that could show some benefit was being taken into patients. So we believe that is what is needed at this stage to be able to combat this epidemic, and we believe that we'll be there at that point in pretty short amount of time. Next slide. I'm going to the next slide, and we already have demonstrated broad spectrum capability. The next step that we have in coronavirus development is that we need to test our drug against the SARS-CoV-2 coronavirus itself. We have a BSL-2 level facility, so we cannot use that virus in our own facility. And we have a collaboration that should be perfected pretty soon. It's a contract in negotiation right now, which will enable us to do the SARS-CoV-2 studies in a BSL-3, BSL-4 laboratory pretty soon. And once we have tested there, then we will shuttle down to coming down to maybe 2 to 3 candidates that we want to take further, do some safety analysis on them, animal efficacy analysis on them to be able to get ready and then manufacture. As I was alluding to, there are a few accelerated first-in-human type of programs that are possible. That does not mean that they are being done automatically. FDA accelerated existing drugs that have safety profiles established in at least in tox package studies in animal models and possibly some human data. They have accelerated those, whether they were tested against the SARS-CoV-2 or not, without efficacy testing. And that's the only drugs that you see currently in clinical trials. Some of them were post hoc tested in cell cultures in SARS-CoV-2 as well. there is no effective animal model as such that produces clinical manifestations in the animal of the SARS-CoV-2 disease as it is seen in humans. But we have gone ahead with an animal model that is reported by Barrick Group, which was used for remdesivir, the Gilead drug, and we have done similar study with that, which will be employed, which we will be doing just to develop the model that it can be used without the clinical manifestation, but with certain metrics that you can use. The viral load can be used, the weight loss can be used, that kind of thing. So we have an animal model in development that we will be using for once the cell culture data in the SARS-CoV-2 model itself, the virus itself, shows which particular candidates we should go ahead with. So that's the update on the coronavirus. We actually do have a drug candidate that can go further. We have, fortunately, we have several of them that are showing similar effectiveness and very, very high effectiveness in cell culture models. We will be starting to do some, we are trying to put together a journal article actually on that right now. And so the data is going through our scientists for reviews. Multiple reviews are needed for this kind of sensitive data sets, and we are doing that instead of just going into a press release. But we believe we can give confidence to people that, yes, there is a drug that is coming through. And that much I have confidence in this data set. And of course, we are going to wait for the SARS-CoV-2 data set as well. And once that is there, we can certainly say that, yes, we have a drug for this pandemic. Going back to what we were working on before, the shingles drug, which is our lead drug candidate, NVHHV101, formulated into a skin cream so that we can apply it topically where the shingles rash happens and where you can provide the maximum drug dosage because you are actually applying it directly on that. The current drug that is available for shingles does not work very well. It's a acyclovir analog called Valtrex. It's given orally, but in very, very high doses, and the actual concentration at the site is not sufficient to cause a major impact, although it does has given some clinical improvement. It is not major. So our clinical studies will be with standard of care, meaning Valtrex will be given. In addition, our drug will be applied as a skin cream. So with that twofold effect, we believe that there should be a very strong effect on the shingles case, patient relief. And we are hoping that it can reduce the progression of the rash and reduce the time that the patient spends with the shingles rash. So that the clinical trial design is being worked out right now, and clinical site selection is also being worked out. So once those two things are done, we can file an IND. Our manufacturing for that was completed, like I said, in the preclinical package, tox package studies. We had already done a cGMP batch. We have now getting ready for cGMP batches, additional batches for clinical trials, and then came along the COVID-19. So with the same polymer backbone, we shifted to developing a drug for COVID-19. And because of that, I can confidently say to you that yes, we can make the drug in large quantities with no issues, with no additional time lags that are needed in further development of CMC and manufacturing controls and so on and so forth. So with the shingles rash, I already alluded to that this is a broad spectrum drug as well, and it has many indications arising out of the herpes virus family. Two additional indications are skin cream type of applications, which is for the cold sores caused by HSV1 and the genital ulcers caused by HSV2. And so once we get these clinical trials rolling, we intend to get started on the HSV1 and HSV2 programs in full stream and get those towards clinical trials. The tox package is done. If we do a variation on the drug, we'll have to do some additional things. Obviously, whenever you need to go into a new drug application, you do want to go with the best foot forward. So we may do some additional variations that will depend upon how the animal studies work out for the HSV1 and HSV2 programs. And the animal model for that, for at least for HSV2, University of Wisconsin has told us it is in usable state now, and in the sense that it will be able to tell if a drug works better than acyclovir. And that was our major constraint, that the current animal models in use could not distinguish a drug if it was superior to acyclovir. And as you know, we would want to have superior drug rather than something that looks the same, because if we have superior drug in animal model, then there is a very good chance that we will have an approvable drug in clinical trials. So that's where we are going to. Shingles itself, we have done market studies that have demonstrated that it has $1 billion market in spite of the Shingrix vaccine that is being rolled out. The dosages being manufactured on that vaccine is currently the manufacturing capacity is somewhere about 7 million doses per year, which corresponds with 3.5 patients per year or persons per year, 3.5 million persons per year. And obviously that means you're talking about 100 years of treating Americans alone. And that means there is a tremendous amount of market that remains available. No, I mean, you don't have to give the vaccine to all the population. So I'm saying maybe 20 years is better estimate for saturation of the market, but there is plenty of space there. And we don't know what will happen with that vaccine afterwards. As we do see vaccine resistance coming in once vaccine applications go into large dimensions of usage. The HSV1, HSV2, cold sores, genital ulcers, these are multi-billion-dollar markets. As you know, HSV1 is highly pervasive, probably about 60% of the population, I believe, and HSV2 maybe about 10 to 15% of the population, but it is a sexually discriminating disease and causes tremendous amount of social issues for the patients who have it. And so those are very, very important diseases, both in terms of helping the patient as well as in terms of economics. So that's the update. And in terms of the shingles, the current progress, we are already there. IND drafts are being circulated. The safety toxicology studies were highly successful. We had to go to very high dosage levels, the maximum feasible dosage levels, and we still did not see any toxicity effects. And we have retained clinical trial design consultants. We have retained consultants for being able to help us with discriminating between different clinical sites and clinical operations. As you know, we are an R&D company, so we did not have the clinical expertise in-house, and that we are bringing in through consultants. We have also retained consultants for regulatory affairs, for going through the IND drafts and making sure that we have the information that will be necessary in there, and making sure that the work is all done appropriately. So that all has been done. We are developing, and we filed a pre-IND application and had a pre-IND response from FDA before for a phase 1/2 adaptive clinical trial protocol, wherein we will do safety and immediately slide that clinical trial into efficacy, at least the initial efficacy studies in humans with disease. So that will have a lot of important connotations there in terms of both contracting the timeline as well as reducing the cost of the trials. So I already talked about what we do, that we go after the receptor on the cell surface and we try to mimic it. So this slide that you're seeing shows a schematic of that HIV virus binding to the cell. This was a figure from the CDC actually, and a published paper in the Cell long time ago. And it's showing the virus, HIV virus binding through its GP120, the trimeric things that you see, club-like things that you see, to the CD4 protein on the cell surface. And what we are showing in the schematic is that we take that footprint portion on which the GP120 is binding, and then we come up with small chemicals that mimic that, and we show that through different studies, one in molecular modeling and then later in cell culture studies, that those small chemicals are indeed effective as ligands binding to the virus. We attach those chemically to a polymer material which looks like a comb, basically a hair comb, except that it is highly flexible. So think of that hair comb as collapsing on itself, and the teeth going inside, those are fatty chains, and the surface becoming PEG, PEG, polyethylene glycol. That's a molecule that is commonly used for attaching to antibodies so that the antibodies will not be rejected by the body when antibodies are being developed as drugs. So that process is called PEGylation, and we use that PEG molecule to begin with as the backbone of our polymer. So that gives us, and that is why we have shown and seen very high safety levels for most of the drugs that we see. Of course, the safety will depend also on the safety of the ligand, and if we choose a bad ligand, we will not have safety. So a lot of thinking goes, a lot of work goes, a lot of development work goes into selecting the ligand as well as the polymer backbone. And the polymer backbone defines what way we are going to introduce that drug into the human body. So for the skin cream, we made a polymer backbone which was more oily so that it can penetrate through the skin, it can go through the skin pores into the place where it needs to go to. And for injectables, we had previously made polymers which are more water soluble so that they can be injected. For orals, there is a different way, but it is all at the polymer design itself that it is encapsulated in the polymer design. This is a tailor-made technology where we make the drug based on constraints of target product profile right at the beginning. Most often, drug development fails at a very, very late stage because these constraints are applied at the end stage. And there are many, many examples. There are current clinical trials going on. There are some examples in that as well. So the other view of how the nanoviricide works against the virus, and in the next slide I will show you electron micrographs which actually match with, look at the number 2 here, and one of the electron micrographs actually matches with that. So what we think happens when a nanoviricide binds to the virus particle is it's a Velcro tape type of attachment. So one loop and one hook interaction is very, very weak. They can come apart. But there are many, many hooks exposed on the nanoviricide surface and many, many loops there on the virus surface. So because of that, a single interaction leads to multiple interactions from 1 to 2 that you see in the slide. And then what the molecule the polymer that we have made is a flexible polymer. It is not a hard particle. And the advantage for that is that, like I said, the lipid chains going inside, now it is seeing the lipid surface of the virus, and now the polymer senses that and it flattens itself onto the lipid surface of the virus, merging the lipid of its interior into the lipid coating of the virus. When that happens, the virus is destabilized because viruses are naturally unstable species. And the glycoproteins that the virus requires for binding to the cell, they essentially get uprooted. So that's what happens in our case. I think I should be stopping with this slide 12 that is coming up, slide 13 that is coming up, just to explain to you the technology. And here in the slide A, you see that intermediate structure where the unstained area on the left top corner, that is where we believe a nanoviricide type attack occurred. And what happened in this particular case is that it pulled the lipid surface of the virus together into the nanoviricide. And that's why you see that one unstained surface. And the interior of the virus, in this case, it contains multiple capsids, and they just streamed out. And in C, you see just the streamed out virus capsid particles. So we do see that this mechanism can work and has worked, and we do see that clinically it works. And now we are progressing to show that it works in humans in the clinical studies. And once that is done for one drug, that is the end of everything. That means that it works for entire platform. And you can see how many different things we have been working on in the other slides. I have to stop here because my time has been up for 10 minutes ago. Thank you very much.
Thank you. This concludes today's webcast. You may disconnect your lines at this time. Thank you for your participation.